Hello, I'm Andrew Price from 10x Genomics, and I'm here today to talk about a scalable solution we have for calling copy number variations on individual cells. This product uses the 10x platform, and so we start with individual cells or nuclei, either from cell culture or isolated from individual tissues. They are entrapped in a microfluidic platform into a gel matrix, which gelates into what we call as a cell bead. The cell beads allows us to process each cell individually to unpack it from its chromatin structure. Each cell is then partitioned into a second partition, which includes a 10x gel bead, which allows individual barcoding of each cell. That cell is then coalesced into a single phase, which allows us to complete our 10x library, which is sequenced onto a standard Illumina platforms, which allows us to call copy number variations with a turnkey pipeline and visualization software. We can start with a variety of input cells, including fresh tissue, frozen tissue. Fresh tissue allows us to either use dissociated cells or allow us to isolate nuclei from them. Frozen tissue, we can get nuclei isolation. We can also use cultured cells or primary cells in this system. It has a very low doublet rate, which is essentially following Poisson loading. And so 1,000 cells generally has a doublet rate of under 2%. The chemistry allows us very even profiling, which allows us to call individual copy number variations with minimal wasting in our sequencing. Our platform allows copy number variation calling on individual cells, generally at the depth that we recommend sequencing to. You can call it to achieve a resolution of around 2 megabases on an individual cell. However, with clustering, we were able to achieve individual resolution of 100 kilobases per cell. Here we show two references, either cosmic or one that we generated in-house from running PCR-free TruSeq libraries and using the Ginkgo platform. We're using a gastric cancer cell line, MKN45, to call the individual copy number variations. To go into a real sample, we have a tumor, which we macro dissected into 5 different pieces. Overall, we achieved over 10,000 individual cells that we sequenced of around 50 to 200 megabases per cell. Looking at the entire tumor, we were able to identify over 5,000 cells which had copy number aberrations in it. And further looking into that, we were able to identify 9 individual subclones that contained over 80 cells per clone. As we can see, looking at each individual sections, 1 through 5, is that each section varied not only in the amount of cancer cells that were detected, from 9% in one section to over 82% at a different point in the section, but what becomes clear is that if you look at the heterogeneity in the clonal structure of it, the amounts of each clones varied tremendously from each of the individual cells. So for example, if we look at clone A, we can see in one section, it represented a very small part of the cancer cells that were there. We can see as we proceed through the tumor that A clone grew and to become a very large section in sections 3 and 4. However, in section 5 again, that became a smaller clone. If we look at the other clones, A through I, we can see that they vary as we go from each different section. And so for color copy number variations, the platform allows us to achieve, at the recommended depth, 2 megabase calling on an individual cell. However, in clustering cells together, we're able to achieve calling on a resolution down to 100 kb. And finally, the platform allows us to interrogate real samples, as we're showing here, in this case a breast cancer tumor, and allow us to measure heterogeneity that exists within the tumor on a very easy and scalable solution for identifying thousands of cells at one time. Thank you for your attention, and if you have any further questions or want to learn more about this product, please visit 10x Genomics website. Thank you.